Right. Hello, everyone. It's 10 o'clock. This is Mark Leefring from the New Zealand Ag Emissions Centre. Welcome to this second seminar or webinar in the series on soil carbon. My co-host is Felix Siegel from the Tunin Institute, and these series of webinars have been organised by the AgriDens initiative. Just to say, organising these multi-time zone webinars are quite difficult to accommodate everyone. And I'd really like to give a shout out to Europe, who are doing the hard yards today. We're nearly midnight, so no, thank you for all attending. So today's webinar is on, sorry, I'm just trying to share a screen here. If I click that button enough times, again, is on analytical approaches for characterizing soil carbon. As I mentioned before, it's been brought together by the AgriDens initiative. AgriDens was set up, a year or so ago, and it's a dialogue on climate and agriculture between New Zealand and Germany. And it's really to foster understanding and political and scientific cooperation, and not just between New Zealand and Germany, but also with third country parties. In the broader initiative, there's 4 pillars. There's farm level greenhouse gas accounting, food loss and waste, and third country engagement in livestock, and the most important one, the soil organic carbon in agricultural soils pillar. So the setup for today is we're in the middle of this welcome by myself. We've got 15 minutes from Marcus from the Tunin Institute, and Pierre from the Bioscience Institute Manaka Whenua from New Zealand. We hand over to Sanani from CSIRO in Australia, and finally Virginia from INEA in Uruguay. So each session will have 5 minutes of technical questions, which you can address in the Q&A button tab, whatever you want to call it. And we'll have 15 minutes of general discussion at the end. So just some housekeeping. So all participants except the presenters and hosts are muted. And like I said before, you can ask questions via the Q&A button. Felix and I will read them and present them to the presenters at the end of their session. There's time at the end as well for general discussion. And this webinar is being recorded and will be uploaded to YouTube, and the link can be found at the AgriDens website. Have I missed anything, Felix, or shall we kick off? No, Felix doesn't seem to be able to say anything, so let's kick off with Marcus. Could you please share and give us your presentation, please?
Yes, I'm just sharing. So I think you see my slides.
Yeah,
great. So hello everyone. Good night, good evening, good day, wherever you are. For me, it's good night. So I want to talk about the principles and application of mid-infrared spectroscopy on studying soil organic matter composition. And we thought, because a lot of this webinars on spectroscopy, that I start with a kind of introduction to the general principle, what it is. So before we go into how to apply it, I just want to start with what is mid-infrared spectroscopy in general. So get through some basics so that we are all on the same page and understand the technology. So mid-infrared is a specific energy range, so it's a part of the light, as we all know. So we have light from 2,500 to 5,000 nanometers, and we mostly express this in mid-infrared spectroscopy in wave numbers. So we call it between 4,400 centimeter or centimeter inverse. And this is the mid-infrared range, so we are in between the near-infrared and the far-infrared range. And what is important, this is a radiation, so we have an energy flux, which is also then expressed here in electron volts. So this radiation, if we point it on a sample, for example here, a molecule of water, then this radiation will turn the functional groups of the molecule into movement. And each of this movement kind of needs a specific energy for each of these functional groups. So for water, we have already 6 types of movement that the atoms can move. So the most common is the stretching and the bending. And then there are several other movements for this molecule. And this is a quite important information because this is kind of what we measure, because this movement that is created kind of transfers energy from the radiation from the light into kinetic energy, means that it's less in the light, which is what we measure. So we measure absorbance. The light, the sample absorbs part of the light to create this kinetic movement of the functional groups. This can be done by different technologies. So there's transmission FTIR, there's attenuation reflected ATR, and reflectance FTIR, and drift spectroscopy. And this can be further split apart into different types of reflectance FTIR. And FTIR is a Fourier transformed infrared technology. It's just a type how it is measured, that every wavelength is measured at the same time and mathematically transformed, that we get one spectra of absorbance. And the type of these FTIR techniques just describes as the light goes through the sample. ATR means the light goes through a crystal through the sample, and reflectance means the surface of the sample is reflecting the light. These are kind of the basic principles for mid-infrared spectroscopy. And the most common or the most advancing technology that is used in soil science is diffusive reflectance infrared Fourier transformed spectroscopy, so the drift mid-infrared spectroscopy. And for this, we have just a rough surface, which is emitting the light again, and we collect the diffusive light, which is coming from the sample. So in more detail, it looks simplified like this. We have a IR source, the light is forced to the sample, and then the diffusive light is collected again and goes into a detector. And this has the advantage that we can use the sample as a powder, so we don't have big sample preparation. For the other technologies, we need to prepare the sample in specific ways that they are in flattened surface, that they fit into the holders, and so on. And the disadvantage of this technology compared to the reflectance and transmission technologies is that we lose a lot of energy due to the rough surface. So some of the light is actually going into the sample, not going back out of the sample, or is missing the mirror, which means that we have less energy that we can actually measure, less light where we can measure the absorbance in the end in our detector. And now we have effects which we need to consider. So these are the effects of water and CO2. So mid-infrared is also used as detectors for water contents and CO2 contents and other spectrometers or analytical devices. So we need to account for this when we look at mid-infrared spectroscopy. And this is just where water and CO2 is occurring within our spectra. So these are the wave numbers. And when we now take a spectra, for example, of a soil sample here, we see that there's a lot of noise. So this kind of needs to be corrected out there. And for this, we need backgrounds which have 100%, nearly 100% reflectance of the mid-infrared range, so that we know what is kind of in the whole machine in the atmosphere, that we can take this out from our measurements. And there's different types to do this, and we use different roughnut gold here, which has a full reflectance, and in our laboratory we use this now as like a standard. So And I forgot to mention, I'm at the Thünen Institute and in charge of the German Agricultural Soil Spectral Library. So most of the work I will present today and the procedures with using the gold is what we do in the German Agricultural Soil Spectral Library. So now we know the basics, and now the question is, what is in the mere spectrum of a soil? And it's simply saying a lot. So when we now look at soil spectra, and this is just, I created this figure based on the German Agricultural Soil Spectral Library. So this is just average spectra of many thousand spectra I have in the library, or we have in the library. And there are distinct regions we can look at. So there are regions where we have absorption of single bonds, where it's mainly water, and also OH bond of clay minerals, and aliphatic bonds, CH bonds. We have region of triple bonds, double bonds, where we have aromatic bonds, amide bonds, and also quartz and silicate. And then we have a fingerprint region in the high energy region, where we have many different absorbance of also aluminum silicate, CN, silicate again. So you see that we have a mixture of everything across the whole spectra, but very unique features that we can work with. And a common figure that is shown is this from a paper from 2015, where it's kind of nicely summarized what you actually see. So there they put also the names of what you see. So you see kaolinite peaks, gibbsite peaks, aliphatic peaks of organic matter, calcite, and so on. And we see this is quite a diverse mixture. And when you get used to spectra, you see actually quite nicely the fingerprint of different soils. And you see this in this spectra that just by looking at the spectra, you see what is the dominant feature in the spectra. So for example, this one is dominated by kaolinite, has this very distinct peaks here. We see a very typical picture for spectra for organic matter, and we see typical spectra for quartz dominated. So this is what I like in this method, that you actually see in this curvy lines you get from your powder what is actually in there quite directly. And now let's move to organic matter. So we look at functional groups of organic matter, and there are long lists at which wave number we have what kind of functional group and what kind of bond. And as a kind of start paper to get into the method, I really recommend the paper from Andrew here, where also this list is from, where it's really nicely shown what are the basic steps of spectroscopy and how it can be used for soil organic matter analysis. Now the question is, we have this list of many different bonds and at which wave numbers they have their absorbance, and several of these bonds have several wave numbers because they have this different types of movement, which I showed before. So there's a stretching and bending of the similar bonds. So a CH bond, for example, occurs at many different locations in the spectrum. So now the question is, if we have this mix of everything, how can we determine organic matter composition in there? And I want to just briefly go through a few examples here. And I want to start with just looking at organic matter without any soil, so pure organic matter substances, how this looks like. And for this, I want to just take a study which we published on the effect of different root quality on soil organic matter decomposition. And to understand what is actually the composition of our roots, we used mid-infrared spectroscopy. So we had roots from miscanthus with different CN ratios, so different contents of nitrogen ranging from CN 50 to 124. And we see that there are distinct differences here in the spectrum. And now we took just individual peaks of the spectra. So this is just a zoom in of this part here, and looked at the relation of these different peaks to each other. And in this table, you just see the composition, for example, aliphatic to aromatic bonds, polysaccharides to lignin typical bonds, and their ratio together with the CN. And this highly correlates with the CN ratio of the sample. And this indicates and showed us that actually our nitrogen-rich parts of the plants in our experiment showed that the nitrogen is stored in the lignified and aromatic bonds. So this is just one application on a pure organic matter that we measured with mid-infrared. And then we can also use it for other types of analysis, so not just looking at the spectra and visually compare it. We can also treat the spectra a little bit. So one way to do this is spectral difference. This is a technique that can be used if you have the same sample, treat it in a certain way, and then scan it again and then subtract the two spectra from each other after some normalization work and technical details. But in principle, what you see then is a shift in functional groups in the sample. And here's an example from pyrogenic organic matter, so fire-derived organic matter or biochar in this case, so artificially produced. And we oxidized this to mimic aging of the char and studied then the processes in the soil. And here was just to prove that our oxidation actually changed the functional group of our char surface and therefore the reactivity of our sample. And you see with this, when the number is positive, that we have a higher oxidation. And you see with mid-infrared, we could clearly show that this oxidation worked and that we have a char which is now more enriched in oxidized compounds. So this is just another way to use it. So this really requires that you have the same metrics and make the assumption that nothing else in the metrics change, which will affect here the spectra. So then there are ways to actually look at the relative composition. So rather than now looking at which wave numbers goes up and down, we can look at the relative composition of organic matter. And the idea here is that we take peaks of interest and we can take peak height or peak areas. I prefer peak areas in most cases, and derive proxies that represent some biological processes or biogeochemical relevance of the compound changes. And it's important that there needs to be normalization, so there needs to be some background technical work, which I don't go into detail here. And what is more important is that we need to have the understanding that we look at relative difference between the sample and not absolute amounts. And what I mean with this, I just want to show this example from a former colleague from me, from EJ Zurich, where he just showed very easily in this graphic what we are looking at. So for example, soil A and B. Soil A both have aliphatic and carboxylic compounds, and we take the ratio of the peak area we observe. We get a specific ratio, and then we, for example, can say in this case, we have a decrease in the ratio, so a higher relative contribution of carboxylic compounds. And when we assume that this is an indicator for processed organic matter, we can say that soil B is more processed. The organic matter in soil B is highly or more processed and more transformed than in soil A. However, we cannot say that soil A or B contains more of the one or the other compound. So it's really just the relative contribution and relative composition of the organic matter. And this is a quite simple approach, which we pushed a little further, and I want to go into detail of this. So this is just, again, how the German Agricultural Soil Spectral Library looks like when we put all the spectra together. So nearly over 6,000 spectra and shown here. And now we look at the spectra in these ranges I just mentioned. So aliphatic ranges here at 2,900 and aromatic carboxylic ranges at 1,600, and the zoom into this region. What we can do now is to take the local baseline and extract the peak areas of these. And we can assume that the aliphatic can represent something which is fast cycling organic matter, it's plant derived. And then we have slow cycling, which is aromatic carboxylic. And this, we always have to keep in mind that there's a restriction of mineral interferences, but these two regions turn out to be meaningful to extract actually directly from soils. The proof is that it works in models. So Moritz Laub implemented this into the DAISY model, just the two pools and the peak areas I showed, and this worked to improve the representation of microavailable soil organic carbon in bare fallow experiments. And more recently, we applied this approach also to look at land use change sites. And when we look at carbon changes with land use change, so different land use changes, and these ratio of the fast to slow cycling And in comparison to different methods, so the blue is the thermal method, particle size fractionation, and mid-infrared, we see that there's quite a high sensitivity. If we have a lot of fast cycling organic matter, so the aliphatic, we had huge losses of carbon and larger increases if our organic matter is enriched in already processed organic matter. And this method comparison we presented or published just recently in Geoderma, so you can go and check this out. And now I talked about mineral interferences, and we have these two regions which seem to be very safe to use also across soils. And their approach is to actually get to organic matter spectra or enriched spectra from soils to look at this across different mineralogies. So what we do here is systematic assessment of organic matter removal and spectral subtraction. This is a way to actually get into the individual spectra that are within the bulk soil spectra, so the mineral enriched spectra and the organic matter enriched spectra. And the idea is actually we scan the bulk soil, we remove organic matter, we scan the mineral enriched spectra, and then we derive the organic matter spectra. We selected here 6 different soils, different carbon contents, which you see in the different spectra. Then we have the mineral enriched spectra after organic matter removal, so testing different methods to optimize this process. And then we can subtract this by zeroing out specific region, and we get organic matter enriched spectra. And this is quite promising and work in progress. So we see actually when we plot these spectra into dimension reduction space, or with a PCA, we see that this is clearly separating between bulk soil and the mineral spectra from the different soils, and organic matter is separating nicely from the two and very close to each other, showing also similarity in the organic matter across the different soils. So this would allow detailed analysis of composition across soils. However, there are also artifacts here, because it's not so easy to disentangle this complex matrix of soil. Plus, spectroscopy creates something non-linear, so applying this operation there always creates artifacts which we need to consider, but it can be a major step forward to actually use spectra more directly. So to sum up this very quick introduction to mid-infrared spectroscopy, I hope I was able to show that we can create compositional data based on functional groups from mid-infrared spectroscopy to characterize pure organic matter pretty easily. And we have to think about the interference of organic matter and mineral phases in soil. However, we can get some relative composition that aligns with biogeochemical relevant processes, and spectral subtraction can be useful, and some work in this direction needs to be done to use it more efficiently. And more importantly is actually that this is all quality, and quantitative analysis need more advanced modeling, which will be part of a next talk in this session. And with this, thank you for your attention, and happy to answer some questions and for the discussion.
Thanks very much, Marcus. There's no questions in the chat at the moment. I just have a very high-level question. What's the cost of these analyses you're talking about in terms of the machine itself and the cost per sample?
So the machine itself depends on the producer. So there are several, but it's roughly in the region of 50, 60, 70,000 euros. So everyone has to calculate this in their own currency. And the cost per sample is very low. So it is less than 2-3 euros with all the operational costs. So yeah, internally we calculate actually 60 cents per sample, because there's no consumable you need. So when you have the machine, you not just need your powders, and then it goes in there, and as long as the light source is stable, it works.
And someone's just saying, sorry, do we use Q&A as the chat is disabled? Interesting. Okay, we'll try and fix that, Ben Smith. Yeah, we'll try and fix that on the go. No, it doesn't seem to be disabled. Okay, there's one question from Michael. Can you talk about the sample-to-sample variation / error across the field, and how spatially heterogeneity is represented?
Yes, so the error between samples, so sample replicates is very low, but I talked about laboratory-based spectroscopy. So if we go for proximate sending in the field, then we have huge issues with variability and stable spectra. So this method actually allows you to measure a sample that you took, which is dried and milled, and then you measure it in the lab, and you can derive all this information from this. But going for the field measurement itself, it's a big step to go, because there are some really big interferences and uncertainties with water and coarse parts of the sample that will create a huge error in the spectrum. But if you have a milled sample, so just to give an example, we scan in the German Optical Soil Spectral Library, we scan every sample twice. And if I do the comparison with all my 6,000 samples I have right now, more than 6,000 samples I have right now, they're like 10, which I should remeasure. So it's if you have a good protocol, it's really easy.
Okay, there's a couple more questions in the chat, but we'll hold them until the end, just so we can keep to time. Thank you very much, Marcus. Pierre, you're up next.
Yeah. Morena koto katoa. Good morning, everyone. Guten Abend. Buenas tardes. I think I got everyone covered there. My name is Pierre. Pierre Roudier. I'm soil scientist at the new Bioeconomy Science Institute, and sort of part of the Malarkey group as part of that. So today I'll sort of pick up from where Marcus left and talk about those quantitative approaches, both in terms of doing predictions, and I'll focus on soil organic carbon fractions, and I'll explain what they are, but also the mapping, because everyone's excited about maps. And I should apologize to Mark and the team. I did put thanks to NZAGRC rather than Ag Emission Center. I think we're both in changing names, so I'm sure they can relate. But those results I'm going to show, are largely thanks to two NZAGRC funded programs. So thanks to them for supporting our vision. So let's talk about how about soil carbon and how does it get in the soil. So you have a sort of fairly high level pictures of plants essentially acting as pumps, right? Through photosynthesis, they absorb CO2 and they exude carbohydrates through the roots, and that gets into the soil this way. There's actually quite a nice introduction to soil carbon by Lavallée in the conversation, which I've put on the bottom right here. I really recommend checking it out. It's a nice general public friendly introduction to carbon fractions. Now, is it really that simple? It's not. It never is. So if we look a little bit more in details, what's happening once those plants have gone through their photosynthesis and things start to get into the soil, we can find different ways the carbon is actually stored in the soil. And I think Virginia will touch into more detail into it, so I'll keep it very simple. We tend to distinguish carbon that at a sort of fast cycling lifetime on the left, particularly organic matter. They're kind of free flowing in the soil. They're accessible to microbes to respirate and consume that carbon. And on the flip side, you got mineral associated organic carbon. That's your slow cycling carbon, in much more stable form. So two different forms that have different residence time in the soil. And it's an interesting dilemma, because if you put this aspect in the wider context of soil organic carbon sequestration, basically absorbing more CO2 to sort of keeping in the soil, it poses all sorts of questions, because essentially soil is not really a bank for humans to use and store the extra carbon. It's also an ecosystem that needs that labile carbon to function. So we need to find that fine balances between storing organic carbon in a stable form, preferably, because we want that carbon to be nicely locked. But also, we want to enjoy the other ecosystem services provided by soils, and often they need that sort of more labile form. That's something to keep in mind. Now, how do we quantify those poles? So, there's different fractionation techniques. Most of by far the wider method to fractionate carbon in soils is the one on the left, physical fractionation. And we separate particulate organic carbon and mineral associated organic carbon. The top one is the labile form, the one that cycles over 30 years or so, 30, 30, 50 years. And the one at the bottom, mark, is the stable one. That's something that stays in soil much longer because it's physically protected by mineral particles. I'll touch base on a new fractionation technique. We've worked with Marcus on a joint project on thermal fractionation, which takes a different lab technique to fractionate those soils, in fractionated active and stable organic carbon forms. That's based on a thermogram. What's interesting in this study is that the machine that does that doesn't exist in New Zealand. We simply don't have the gear, right? So I'll talk about how spectroscopy can make up for that. Keep that in mind. We go a little bit deeper. That's an illustration of why we're interested in different ways to fractionate. So we can see that on the left here, particulate organic carbon. I'm trying to use the pointer like Marcus. There you go. So we got on the left here particulate and mineral associated carbon, and sort of the colors are that sort of mean residence time. Long story short, physical fractionation doesn't really separate those centennially active and centennially stable fractions very well. There's a bit of an overlap still, while rock eval and that sort of thermal fractionation with a dedicated model can separate better those labile and stable form. So there's the hope that that thermal fractionation will separate those two pools a little bit better. That has gained some tractions sort of over the last 10, 15 years, right? And there's been sort of an understanding throughout the scientific community that there's more to it than total carbon. For a long time, we were sort of focused on basically carbon accounting. This is hard enough to try to understand, how much carbon's in the soil. But now we also want to know what sort of carbon's there. Now, how to scale up those fractionation measurements? I said that thermal fractionation doesn't exist in New Zealand already, and physical fractionation takes a bit of time, and we're just starting to get that ball rolling. So this is where spectroscopy is useful, right? So we've had a good presentation of spectroscopy, of the basics of it. So I'll skip through that very briefly, but basically we have a machine that records reflectance of soils in many different wavelengths in the mid-infrared part of the EM spectrum, right? And as Marcus said, it's fairly cheap and fairly high throughput. So we can process a lot more sample that we could with a sort of wet lab method. We've started quite a while ago in New Zealand. If you look up that paper here, it documents the soil spectral library for New Zealand. This is a little bit outdated now, and that map is now a lot more populated. And we have more than 13,000 samples, individual samples scanned with MIR across New Zealand, and that's growing every day. That's sort of background effort of scanning soils. So, unlike Marcus, we presented sort of that qualitative analysis. We want to predict stuff, so we need to use statistical inference, and to do that, we need a model, right? So we have this sort of collection of spectra that all have reference measurement. And we use statistics really and machine learning and things like that to predict those reference measurements based on that calibration set. The sort of traditional tool is partial least square regression, which sort of reproject, so it's a projection technique finds what's called latent variables. So of course, those thousands of bands, there's a lot of correlation in that signal. So that's a method that allows us to simplify things a little bit and find latent variables that are mutually independent, and also reproject the outcome of the regression. That's kind of a detail though. And then there's just a linear regression between those latent variables. The little fancy stuff we do is to, rather than using a linear model, we use a GAM regression on those PLS latent variables on the scores. We do that because of two things. A, it's a sort of framework, GAM, that can handle nonlinear relationships in the data, but more interesting in my mind, it's also there's some Bayesian approaches that allows to give a full description of the uncertainty And we found that describing the uncertainty to our users is quite important. So to cut a long story short, that's a method that can give us a prediction, but also how wrong we think that prediction might be. So leveraging the existing samples of the National Soil Carbon Monitoring System, we were able to do some analysis of the fractions. And I'll stop here for a moment because I know my colleague Caroline Headley has seen her name, and that's very much a legacy of her work. We established that National Soil Carbon Monitoring System of 500 locations throughout New Zealand. And it's been incredibly enabling for a huge amount of science, including what I'm about to present here. So we have those, about 3,500 samples that have already been samples throughout the country, and we're able to pull out 302 samples for fractionation analysis. What's key here is that we were able to tap into the statistical representativeness of DNSCM for land use, land soil types, etc. Keep that in mind later. You'll see why it's important. Mostly we focused on 0 to 30, but we also sampled some selected 30 to 60 sites. We didn't have to sample as much because it's less variation in those deeper levels. So we're able to sort of calibrate those models. I just show one about total soil organic carbon, which is usually fairly well predicted. You get the prediction as a dot, and then that sort of 95% prediction interval as showed as a line. Now, for the fraction, here are the results. On the top, you have the thermal fraction, stable and active. At the bottom, you have the physical fractionation, the mineral associated on the left, and the particulate here on the right. So you can see that all results are pretty good. The one that was a little bit more challenging, and that'll be true in the mapping as well, is POC, the particulate organic carbon. And we believe that's very much a function of the statistical distribution of that variable. You can see a lot of skewness in the data. And so we believe that we could potentially improve that by capturing more data that have higher POC content. So it may be just a little bit more work to do and a little bit of more sampling to do. Now, of course, once we use those 300 samples, we can apply the models that we created from them. So we are tapping into that huge amount of samples of 13,000 samples that have been scanned in our library. What you see here in green is those 300 samples overlay with what's a density cloud. So the warmer colors shows where most of the spectra are in this National Spectral Library. And what we found that most of them sort of are nicely overlaid by our calibration set. That means that our the models we derive will be valid for most of the spectral library. We have a little routine that excludes those ones that shouldn't be used with this model. And then once that's done, we can effectively leverage the power of the library, deploy our models on it, and here you have it. So the thermal fraction on the top and the physical fraction at the bottom. This is for 0 to 30 centimeter. So that gives about, on top of my head, about 1,500 locations throughout the country, which would be very, very hard to achieve using wet lab technique in such a short amount of time. Now, this is all well and good, but this is still just points. And in terms of stakeholder engagement, everyone sort of prefers maps. This is where we use a paradigm called digital soil mapping. It's actually not so crazy new idea. That guy, Hans Jenny, published this incredible book in 1943 called The Factors of Soil Formation, which were very much putting soil properties as a function of a bunch of environmental layers: climate, biology, terrain, geology, time, etc. Most of this now can be sort of assessed using things like remote sensing, and the F functions is usually enabled by advances such as machine learning. So we're going towards graded soil information products that leverage that paradigm. Just a note on this: it's been done throughout the world. So the Australian have the soil and landscape grid. It's been done in the US as well. And what we do is to use machine learning, in this case, random forest, to match those points with environmental variables that we have at hand. So you can see a list of these environmental variables that I use to, in this case, map stable carbon here on the right. And In this figure, they are classified based on their importance, how important they are in the stable carbon model. You can see that in this case, total precipitation is the main driver. We can see that amongst all these sort of top variables, they're all the usual suspects when it comes to carbon distribution in the landscape. And that's the sort of scatter plots of predicted versus observed values. You do see more uncertainty, more uncertain models than on spectroscopy, which is hardly a surprise. And the R squares are in the sort of 0.35, 0.4 zones, which is a very sort of classic thing in dimensional zone mapping, I suppose. Again, you can see that POC is a little bit more problematic than the others. And again, keep that in mind for later. But once we got these models, we can apply them onto our grid of environmental covariates. These were at 1 kilometer resolution, but could probably be pushed to about 1 kilometer, 100 meter resolution. So on the top, you have the predicted distribution of stable and active carbon, which is the thermal fractionation. And at the bottom, you get the same result for physical fractionation. Now, we can also try and do future prediction, because some of these predictors, like temperature, for example, they have projections, right? Future projections. So that's something called space for time substitution, where we can substitute our predictors, our baseline predictors, for their future values. So that's layers that are curated by NIWA. They have all sort of projections based on the different IPCC scenarios. So we took basically the average IPCC scenario and projected things like growing degrades. You can see a lot of them are going to be a lot higher. Potential evaporation, transpiration deficit, precipitation, mineral temperature, etc. So all those variables have future 2050 projection, can be substituted in the modeling pipeline, and then we can have future projection for all the variables that we mapped for those sort of baseline predictions. Based on that, we can compute a difference between our baseline and our future projections, right? So which give this sort of projection changes. So, of course, a bit of a word of caution. Something we didn't do in this case is to analyze really how the uncertainty goes throughout that pipeline. That definitely be important, in particular for POC. We know the POC model is not as good as the other ones, but it gives us a little bit of an idea of the geography of what may happen. The other caveat in this example is that in New Zealand, we don't have a future prediction for land use. Land use is one of the predictors. And of course, it'll be critical to see how the land use change in the future and will influence the way carbon is stored. Just to finish, a word about carbon vulnerability. I think these things get more interesting when you start combining them. For example, the carbon vulnerability is the labile over the protected carbon. And then you get into, I guess, more of an interesting geography of the soil carbon, of the nature of the soil carbon. You can see here, all of the allophonic soils, for example, we store a lot of carbon. They're really interesting because they usually very high carbon, but we can see that carbon is often very vulnerable because most of it is labile carbon, or a lot of it's labile carbon. We can push further that story. This is work I'm currently developing with Sam McNally, and combine it with things like the mineral surface area, which basically says how much carbon can be stored safely at any point in space. We have the carbon loading, how much carbon is currently safely stored in a stable form in the soil, and the carbon vulnerability. The point I'm making here is that there's no one map that can tell you the whole story, and you got to consider those different integrated maps to have the full picture. And that's currently the scope of our work in the PRISM project. And I'll stop here. Thank you.
All right. Thank you, Pierre. If you have questions, please write them in the Q&A. And also, I think it's useful if you may add to whom you're asking the questions, in case, for example, you have a question about something that has been presented one or two speakers before. And the answered questions will be found in the Q&A section as well. There you have a little thingy just to be sure that you find it. In the meantime, there are no questions right now. So if you have some, just write them in the text box down there. And looking at the time, I would say we're good if we continue. Yes, nothing pops up. So I would say, Sinani, let's go to Australia. And thank you, Pierre.
Thanks, Felix. Hello, everyone. So I'm Sanani. I'm a research scientist working for CSIRO in Australia. So I will try to give a very good introduction from Marcus and Pierre on the spectroscopy and the applications of the quantification methods. There will be some repetition, but I'll try to avoid much as possible and focus on more applications. Before I proceed, I'd just like to acknowledge the traditional owners where we work and pay my respect for the elders and past and present. The work I'm presenting is on my own. It's a collaboration with multiple colleagues, both within CSR and external. I'd like to acknowledge them for their contribution. So there's multiple interest on soil carbon. In Australian context, there's a carbon accounting scheme which is actually being developed with the participation of the Australian government. So there's a credit scheme linked with the soil organic carbon. And then also there's a target Australia to achieve net zero. So there's an ambition that soil carbon can play some role in this towards this journey. And then also there's a long-term stability and the productivity of the land. So it's beyond carbon credits. So if you look at this particular graph, you'll see that there's a commercial readiness in your x-axis and the cost per sequestering 1 ton of CO2. And these are the different technologies that's been looked to reduce the emissions. And you can see that soil carbon is in a higher technology, commercial readiness with a low cost. So it's not the only solution. It's a solution available with a bunch of other options. Now, why soil carbon fractions? I think there's a lot of discussion about this, so I will not go much detail. But I think it's important to understand that the soil carbon itself is a heterogeneous mixture, which is at the different stages of decomposition, and they interact with the mineral phases, and some are protected by the aggregates. So it's all influence the turn over time. So why fractions? One of the interesting things is that it's difficult to understand as it is in the nature. So if you are a modeler or due to understand the mechanisms, it's easy to disentangle them into like small pieces and study them. And then it has an important role in multiple models, which is used for the carbon accounting and in other aspects of the soil carbon change. And also to understand the soil carbon vulnerability, as Pierre and Marcus discussed, and also the quantify the attainable storage conditions, soil carbon. So I use specifically attainable rather potential, can discuss later. Now, most of the fractionation scheme, there was a really good review by Christopher Caprio from Tunen Institute a few years ago on the fractionation methods. But most practically used is this method where you just fractionate into these particulate amino associated carbon. Now, we call them as, particularly we call them as coarse fraction. And then the mineral associated, we call them as when you divide it into two fractions, side fraction, that's fine fraction, instead of particulate and fine. I'll tell you why. So that's the more general application. But in terms of the Australian context, it's a little bit different. Our fractionation methods at CSIRO has been developed over the last 3 decades with James Kingstead and Jeff Boulder, who used to be my PhD co-supervisor. They were very much in a focus on quantifying beyond these 2 side fractions. So we came up in Australia 3 fractions. We call this measurable soil organic carbon fractions. We call particulate organic carbon POC, humus organic carbon HOC, and resistant organic carbon ROC. Now, the availability of nutrients and then the resistance will be changed with these different fractions. And you might see that why we have this ROC, resistant organic carbon fraction. Now, the reason is that if you look at the composition of the carbon, also generally, so these are like 6 samples collected from a project led by CSRO and other partners 10 years ago called SCARP, Soil Organic Carbon Research Program. And if you look at the carbon content, this is the total carbon content. If you look at the New South Wales 00045, New South Wales 00101, you might see that the carbon content is almost very similar. But if you look at the composition, they're different. So in this 00011 sample, you get a little bit higher humus organic carbon compared to the this particular sample. So what it means is that total carbon will not going to provide information on the stability of the carbon or the storage potential. So we calculate something called soil organic carbon vulnerability index. It's very similar to what Pierre presented. So we divide the particulate, which is a very fast cycling carbon content, by the humus organic carbon plus the resistant organic carbon. Now, why rock? So you might think that, like Pierre's presentation did not much focus on the resistant organic carbon. So these resistant organic carbon expect to stay in soil for like a large number of like millennia, but it's specifically derived from the fire. So if you look at this particular map in your right-hand side, so this is like a rangelands in Australian continent. You'll see that the frequency of fire in some areas in northern Australia, there's a quite large number of frequency compared to other parts of the country. So this fire generates charcoal, which is a part of soil organic carbon that can stay in soil for like hundreds of years. I mean, there are new evidence comes that these carbon also cycle and they can be washed away, they can be moved through the soil profile, etc. However, they can be staying for a long time. Now, these fires are part of the natural phenomena, wildfires, or it can be because of the anthropogenic land management practices. For example, the Aboriginal people in Australia have been managing the land for such a long time, and they use these controlled fires to actually avoid these large-scale fires. So it's a fire is an integral part of Australia. So when we quantify different forms of carbon in Australian context, it is important to quantify the resistant forms of carbon. Now, the fractionation methods, as I said, developed in Australia is based on earlier work by James Kempstad and then later modified by Jack Wardock. So the initial methods, like basically if we were using say less than 2 mm samples, we do physically fractionate at 50 microns. So we call coarse and fine. So rest of the world probably call this particulate organic carbon, and for this we call the mineral associated organic carbon. And then once you have the 2 side fractions, we use a virtual fractionation method using 13C NMR, where you try to quantify the resistant forms of carbon. So we call them as charcoal-like material, because we don't specifically say that it's charcoal, it's called as charcoal-like material. So we quantify the charcoal-like material both in the post fraction, because you probably can see some large particle of charcoal to your naked eye, or even within the fine fracture, very fine carbon, stable carbon. And then we derive POC and rock simply by subtracting the charcoal-like material from the post fraction. We derive particulate organic carbon, which is labile, and then we subtract the charcoal-like material derived from NMR from the fine fraction, and we call them as HOC. And then finally, we add the charcoal-like content, carbon content from the post and fine, and we derive it as a rock. So we call POC and rock. Now, this method is been running in our lab for last almost a couple of decades now, with slight modifications. But more recently, a few years ago, my colleague Ryan Ferguson and myself, we were looking for some alternative approaches to explore and speed up and reduce the cost of measurement. So we were looking, we came up, we looked at for different options, and we went through these thermal approaches similar to presentation, some as an example from Pierre. So we done a slight modification. It's basically coarse and side fraction. and then we just use the thermal method to quantify the stable forms of carbon. So our postdoctoral fellow, Claudia, just submitted her manuscript from this work. So she proved that the method can be reproduced to some degree, the current fractionation method. There are differences. It's not one-to-one line if you plot them. However, the cost and the time is significantly reduced. So that makes this can be applied practically beyond carbon accounting for soil health and other applications. So it's basically, instead of NMR, virtual characterization of the charcoal-like material, so we use the thermogram to quantify the thermally stable ROC, and then thermally stable ROC in the fine fraction. And then we deduct the total carbon, and we derive, we call it as coarse fraction TOC 400, so which is approximately equal to thermal derived TOC. And once you deduct the thermal ROC from the fine fraction, we call that fine fraction T400. I'll explain why T400 later. So that's known as can be approximately equal to thermal derived HOC. They're not fully, it's approximately. And then we add the thermal stable fractions we call thermal derived ROC. So it's basically we derive HOC, and ROC with a very slight modification, and we get three forms of fractions. Now, a little bit more about the thermal fractionation. We have an instrument in our lab, which is it's manufactured by Elementa. There are two methods. These are developed by, they call German standards, like DIN A and DIN B. So the one method is that, its combustion happens under the oxygen. So you have the temperature here. And then once that, at this particular approach, we use a step ramping approach. So we ramp the temperature, and we hold the temperature around like 400 degrees to get a peak. So that's a lay-by form. And then we again increase the temperature, and we hold the temperature around like 600 for some number of seconds, and then you get a peak. That's a more stable form. That's called ROC. And when you come to like around 900 degrees, a remaining carbon, that's an inorganic carbon, will be combusted. Now, what we initially thought that we can use the method B, which is like start with the oxygen up to like 400, then shift the gas from oxygen to nitrogen. It's a pyrolysis, and then again turn back to oxygen. So basically, you start your temperature, ramp up to like a 400, and hold it. You get a peak of carbon. So stable, thermal stable, and then you increase your temperature up to like 900, but you keep your carrier gas as nitrogen, so it's inert. And then you just change from inert gas to oxygen, then remaining any carbon will be, organic carbon will become combusted. Now, I'm not going to go much details, but what we decided is that we will go with the DIN B method because of some of the technical difficulties. And we also did slight modification that some of the inorganic carbon can be start decomposing at a lower temperature. So we use a fumigation method to remove inorganic carbon and run the method B. And the results that Claudia presented in her manuscript shows that there's a really good relationship and there's practical applications. Now, I think this part also will be discussed by Pierre and also from Marcus. So once we get this data sets in Australia, we have around like more than, currently with the new project we just finishing off, around like 1,000 observations of carbon fractions scattered across the different agriculture ecosystems and a little bit around natural ecosystems. So we have to get this across the landscape around the other unknown locations. So we normally in our projects, we scan all the samples from the mid-infrared spectroscopy. And once you have the spectral library and we have the measurements, we can develop a mathematical model like Pierre explained. And once you have the mathematical model, we can use that one to carry out the prediction. So it'll take like, a couple of seconds to run predictions from those measured samples. Some applications, almost 10 years ago, actually 12 years ago, Jack Waldock and colleagues published these national scale models. We've been improving those models significantly over the last couple of years. There's a new improvement of these models. And these models can be used to predict. So the x-axis is the total carbon here, and this is particulate carbon, this is humus carbon and resistant carbon, and these are the predicted values. Now, I must note that these models are predicted in square root form, not in original form. One of the things that we found is that obviously it's get a linear relationship, but when you back translate, it's not the best in terms of the model quality. So we have been trying few modern machine learning methods, as well as some of the pre-processing methods to improve these models in the original form. And we have been getting quite a bit of success on these aspects. I haven't included all the results, but there are ways to actually improve it. Now, once you have these measurements across the landscape, hundreds of measurements, what are the other applications? One of the things we've been really looking at the quantify this something called this attainable soil organic carbon in the fine fraction. So the fine fraction consists of the more stable forms of carbon, like humus organic carbon. We want to look at the loadings of this carbon across the landscapes and see whether there's a gap or deficit that we can further fill if there's a suitable conditions. Now we use word attainable because what we measure is what's available in the landscapes. And we know the landscapes consist of different levels of pressures, and there can be different level of constraints. So we can't call it as like what we measure as potential, because we are not running it. We're probably not running at an optimum condition these landscapes, managed landscapes. So what we do is, like, if you look at theoretically, if your fine fraction mass, that's a silt and clay mass, brought in your x-axis and your carbon loadings, this is like a humus organic carbon, you expect that when the mass goes, there's an increasing positive relationship. And then if you fit like a order and least class, you see like a relationship. But then if you fit like upper line, like quantile regression, you will be able to calculate the deficit. So anything below this dash yellow color line, there's an opportunity to increase the carbon content. And then we assume that there's like a bucket size can be further increased. Now, I'm not going to more much details, but it's basically what we've done here to derive this soil organic carbon deficit is simply you have the site fractionation, and we've done the NMR, which is quantify the virtual types of ROC, the alike material, and then we use the fine fraction mass, and then the derived fine fraction carbon content, and then we calculate the regression, and then calculate the deficit. So there's a manuscript we published last year, which is open access. It's available in Soil Research. You can have a look. So this classic example, this is the real data. You can see that you have the carbon mass of the fine fraction, that's silt and clay, and the carbon loadings, that's a fine fraction carbon. That's milligrams of carbon per fine fraction soil gram of soil. And you have the gradient of rainfall. And then you have the different depths. You can see that, despite you have like, with the depth as well as with the rainfall, despite you have the higher carbon content, higher fraction of fine fraction, it doesn't mean that there's actually increase of carbon. You require to have good conditions in the landscapes to actually fill that deficit. And then once you have these data sets, we use spatial modeling techniques, like here explain like a digital soil mapping methods to carry out predictions. So we're able to get the predictions across the landscapes. And then these spatial maps are available at a 90-meter resolution, 100-meter resolution, can be used for policies and other things to come up with different recommendations. Now, applications of these maps in a project scale or farm scale is limited because they are developed at a very much focus on national scale rather than a paddock scale applications. All right, moving to next application of soil organic carbon fractions. This example from the Australia's National Greenhouse Gas Model, which is used for annual reporting under the UNFCCC. We use a model called FullCAM. It stands for Full Carbon Accounting Model. It's a framework which consists of different sub-models. The soil model of this FullCAM framework consists of famous Rothsey model, very simple first-order kinetic model, but it works for the Australia. One of the key modifications done almost 10 years ago, or 10, 15 years ago, is that how we initialize these models in landscapes. Model initialization, process-based model initialization is very sensitive how it's been done, and it can have an impact on the simulated results. So in Rothsey model, there are 5 pools. So these are pools, these are conceptual pools. We call them as DPM, decomposable material, resistant plant material, biome, humus, and inert. So inert assumed to be non-cycling. All other pools assumed to be cycling, or in the model, it's considered as cycling. Now, what we've done is a very simple modification to this model. Instead of using traditional approaches of initialization, using running this model under constant climate or a constant carbon input for thousands of years to come to like that, so equilibrium. So these conceptual pools were replaced with the measurable fractions. If you can recall, the RPM was replaced with the POC, our measured POC, and then the humus is replaced with humus organic carbon, and inert was replaced with the resistant organic carbon. And we have temporal data sets from a long-term experiment, and we do also have the fractions. So instead of calibrating these models to one total carbon, so these models can be calibrated to these 3 fractions, which is like a much more accurate, because you get understanding about the dynamics of carbon more reflect through these fractions. So Australia is probably the one of the first few countries use these type of models, tier 3 models under the IPCC classification, and also probably one of the few to actually use carbon fractions to initialize models at national scale. How it's been run? As Pierre explained, we do have different forms of maps produced at different across the continent. So these maps were produced almost 10 years ago, but these maps are recently updated as well with the new data sets. We have POC, HOC, and ROC. So these are the 3 forms of carbon fractions, and we replace those carbon fraction maps at each grid location of the simulation to get the initialization conditions. And we have clay content, which is a part of the Rothsey model carbon cycling, and we combine it with the incoming carbon, and we will be able to run this model at a 25-meter resolution across Australian continent. So the Australian government is running this model, and CSIRO is a research partner to improve these models with the time. And I think that's probably it, the applications in Australian context. I'd just like to conclude that, there are multiple benefits of understanding the soil organic carbon fractions or composition compared to, or in addition to total organic carbon. If you look at current carbon accounting methods, most of the carbon accounting methods are focused on total organic carbon, or so total soil organic carbon. I don't think there's any issue of that, but there's a growing interest on measurable soil organic carbon fractions, especially with the mean resistance times can be used to actually provide more certainty about the fixed carbon in the soil. One of the important things is that there are really good research tools, but practical applications are limited because of the cost and time. As I explained earlier, some of the methods that we are currently developing through the postdoctoral work that we within CSIRO, what we find is that pragmatic, cost-effective fractionation methods will actually increase the uptake of these technologies in a real-world applications, and it can be used just beyond carbon accounting, and also used for soil health, landscape resilience applications. That's probably it, and thank you very much.
Thank you, Senani. All right. With respect to time, there's one question I would like you to answer. I think it's a fairly quick one. It looks like the light fire or prescribed burning, a natural biochar or hydrochar process, may contribute to permanent carbon storage. Am I correct?
Yeah, look, some of those carbon, like depending on how they produce in the landscape, they can be cycling. So there's more evidence coming up that some of this carbon probably not be in the landscape for millennia, you know. So because they can, if that's the case, the soil should be like a really black in color as well, you know. So what we found is that there are stable forms, like there are different, even within the stable forms of carbon. Research led by Michael Bird, there's a classification that if you have like more than 6 aromatic rings, it's considered as highly aromatic. So that's probably the thing that we are currently using. What we also found is that we don't have much temporal data being analyzed. So we have limited agricultural temporal data. If you had like across the landscape with a temporal data collection from a soil monitoring network, that would be fantastic to actually look at it. But we also need to acknowledge that there are erosion and other movements of carbon in the landscape. Simple answer is that for the models, we assume that these resistant organic carbon will be non-cycling, but in the reality is that this carbon will have some cycling effect.
All right. Thank you, Senani. And if you have more questions to Senani, just put them in the chat. And now we move to Virginia, and we will hear something about Uruguay.
Hello, good morning, good evening, good afternoon to everyone. It's a pleasure to be here with you, and thank you so much for having us in this great workshop. Are you seeing my screen already in the shared mode?
Yes, looks fine.
Yes. Okay, so as anticipated, I'm from Uruguay, from the National Institute of Agricultural Research. I'm going to talk a little bit about the physical fractions and isotope tracing to assess soil carbon dynamics with measurable pools. I'm glad my colleagues anticipated part of these concepts because it's a lot to cover in a short period of time. Let's see if I can. It's not passing the slides, is it? Okay. So we don't have a lot of time to cover such a big content. So we're thinking about just mention a little bit about soil carbon cycling, present the soil organic matter as a continuum, as in the last years it has been framed. Then showing about this compartmental approach of these different fractions, talk a little bit about the isotopes and show some examples. Well, we only have 15 minutes, so I'm going to be fast. When we look at carbon cycling dynamics, everything starts with the photosynthesis, as carbon dioxide is captured from the atmosphere and retained as plant biomass initially. We have, let's say, a complex systems in agricultural lands and in lands when we have grasslands also, as this plant biomass is used to feed animals sometimes, and then we have these inputs to the soils that are usually plant debris and also animal manures. Once these residues get into the soil, they start decomposing and then are finally retained in the soil as soil organic matter as associated to these clay minerals, and then later in aggregates. So to understand better these processes in the soil, we have to understand that soil organic matter is really heterogeneous. In the last decades, there have been a debate about this process of soil organic matter formation, let's say. We can say that there's lately a consolidated view that we have a soil continuum from plant debris to and animal residues to the final soil organic matter that is stabilized, attached to the mineral surfaces. Plant and animal residues can have complex molecules that then can degrade to polymers and then to smaller polymers to finally monomers to end up in carbon dioxide. At different stages, we can have a process of sorption and desorption from mineral surfaces of the different type of molecules. So it is let's say it is really hard to put this soil organic matter in one compartment, in 2 or 3 compartments. We assume it is a continuum, but we need to find some proxies to understand better how we can approximate then the different time frame for this soil organic matter in the soil. So here it comes this I'm sorry, but I see something covering part of my screen. Probably you don't see it, I guess. In the last decades, maybe the most used type of fractionation is this particular and mineral-associated organic matter fractions. We have these physical fractions separated by size and also by density. This is a nice schematic from Laval and colleagues. There have been different approximation methods, and I just name a few here. We have more. They are all somehow time consuming, so it is great that new approaches appear to simplify this in shorter time periods that we can analyze. But so far, we assume that we have, in terms of size fractions, the typically is to use 2 big size fractions, the mineral-associated organic matter, as we separate in a sieve of 53 microns, everything above the sieve and everything below the sieve we call the mineral-associated organic matter, and the particulate organic matter. Although we know this is not really a perfect separation, we also have dissolved organic matter that is actually cycles in a fast turnover, and it ends up in the mineral-associated organic matter fraction when we only take the sieve. We could separate this by density, although we know it is really time consuming. So then when we talk about isotope tracing, excuse me, I have so some basic concepts. Some basic concepts. Where are the isotopes?
Thank you, Tom, Virginia. No rush. No rush. Are you there, Felix?
Yeah, sorry, I just catch a cold in the last couple of days, and it's unfortunately, I'm sorry about this, but
No, relax. Okay. Take your time.
Yeah, okay. So that for a general introduction about the physical fractionation, I think now we can cover some of the basic aspects of the isotope tracing. So what are the isotopes? Isotopes are atoms of the same element, which differ in the number of protons, but which have, the same number of protons. That's what makes it be the same element, but that differ in the number of neutrons. And so they differ in the atomic mass, but not on the chemical properties. And for carbon, we have carbon-12 and carbon-13, which are the most abundant. In particular, carbon-12 is by far, very far the most abundant. It's about 99% of what we find. And in carbon-13, it's only about 1% of what we have in the Earth crust. And we have carbon-14, which actually is, we have very, very, very little of it. We only have traces of them. The first two are stable isotopes, while the carbon-14 is non-stable. It's being decomposed with time. It's radioactive. It's radiocarbon. So how can we actually use this to understand the soil carbon dynamics? Natural abundance of carbon-13 has been used as a tracer for a long time, and I did put this in this couple of slides of very old works to understand this is not a very new concept, let's say. But where does this derive from? Well, I was showing at the beginning that we have this the carbon that's stored in the soils can ultimately be traced to photosynthesis at some point of plant photosynthesis, and according to these the different photosynthesis pathways of the different type of plants that we have, these different isotopes with different masses have different time of reaction with the different enzymes for photosynthesis, so we can find them through the different pathways in a different relative abundance according to this photosynthesis pathway. So we can track according to the ratio of carbon-13 to carbon-12 respect to a standard, a different signature of the carbon that was first photosynthesized by a C3 plant or a C3 or C4 plant. And we can distinguish that in the soil carbon that we see. So it has one of the typical uses has been to follow up when we have a change in vegetation cover from a C3 to a C4 plant that would be marked in the soil. We can then use take advantage of this to calculate the turnover time of this carbon that has been that has a temporal decay that can be calculated according to the amount of this carbon that we can distinguish from the different origins that we see in the soil. We can do that when we have a change in vegetation from C3 to C4 or vice versa, but we can also do that by incubating samples that we have previously enriched in carbon-13 or in nitrogen-15. This is an example where we used take advantage of this stable isotope tracing to assess the carbon and nitrogen dynamics in soils that had been under different originally soil carbon contents, and therefore we could think that we're under different saturation conditions after they had been under different either cropping or integrated crop pasture systems in Uruguay. We knew they would have different soil carbon contents, and then we wanted to see if we added this carbon that we could trace in these labeled residues, how much of that we could retain in the soil according to this initial condition of the soil. So we went to the field and we sampled soils from different systems, the ones that are perennial system with crops and pastures, and then others that were under continuous cropping in the different across the soil profile, the topsoil and the subsoil. And then we incubated this with different amounts of residues that we had produced, taking advantage of these different isotope tracers that we could use. We ran these incubations for several months, and then we assessed the retention of this carbon and nitrogen following these isotopes, and we calculated the composition and the rotation of soil carbon in these incubated soils in both fractions, in particular in mineral-associated organic matter. So what we saw from these isotope tracing experiments is that as we approached this theoretical saturation of the soil organic carbon content respect to this theoretical value of saturation content according to Hassing and Whitmore equation, as we approached this theoretical saturation value, we could see less carbon retaining the soil as a proportion of the inputs that we were adding. Remember, we added different amounts, and for all of the different amounts, we could see the same effect, even in 2 sites. This is a long-term experiment in La Estanzuela, and that is started in 1963. I'm going to comment that later. And then we have other one that started under no tillage in the 1995, and still we could have the same results. When we track this carbon in the from the residues that we had added and see after the incubation how much of that we could have in our soil mineral fractions. Now, that's to have an idea of what we can do with stable isotope technology. But then just very briefly, I'm going to comment a little bit on this radiocarbon dating that we can use to study the soil carbon dynamics. I was mentioning that radiocarbon is very, very little, that one that we have. The radiocarbon content was increased in the 60s due to this testings that were done at the time. Where if we see the abundance of these delta radium carbon 14, we see there was very, very, very little amount in the atmosphere until this year. That was this peak that is called the bomb testing peak, when this radiocarbon was more abundant in the atmosphere. That actually market all this, let's say, the photosynthesis was done from there on, was taking this tracer, let's say, that we had from there in the atmosphere. If we zoom in the time, we see that this peak occurred after 1960s, and then we happen to have one of these long-term experiments that I just mentioned that started in 1963, in which we had compared continuous cropping system without fertilizer, continuous cropping system with fertilizer, a system that was under continuous cropping and then converted to integrated system with pastures, and then a system of production that was always from the very beginning of the experiment integrated between crops and pastures. So we were wondering which are actually the mechanisms behind this success of the integrated systems, and we can have greater soil carbon stocks when we have perennial pastures integrated in the rotation with pastures. If it was a matter of what is the real mechanisms? Is it that the pastures can increase the soil carbon because of greater inputs, or is that we actually retaining all carbon that we have in the soil when we compare with these continuous cropping systems? So we assess that this by using radiocarbon dating is this in this published paper from Maximiliano González. This was a collaboration with the Max Planck Institute for Biogeochemistry, our National Agency for Research and Innovation, the National Institute of Agricultural Research, the local university, and at the Faculty of Agronomy. So how we did this? Here is Maximiliano working on the experiment. First, we look at the temporal series of soil organic carbon and the productivity of this long-term experiment that we could use as a proxy for the soil carbon inputs. Then the soil was fractionated in particulate and mineral-associated organic matter. And then, so this was measured in the soil samples, and also samples were incubated to see the signal of this carbon dioxide that was leaving the system, to see if it was coming from the stable pool or from the most labile pool. That is, we approximated this with the particular organic matter and mineral-associated organic matter, and put together differential equations for the topsoil layer. What we could see is that actually the soil organic carbon in the integrated systems are very old. The signal of the bulk soil follows very much the mineral-associated organic matter, which is about 600 years old. When we look at how much of the carbon really entered in the system since the experiment started, as we could trace this with the carbon-14 since 1963, only about 8 or 9% of the soil organic carbon entered the system during that time. So this difference that we have between the integrated system and the continuous cropping system, that is about 30% of the carbon stocks, actually is more related to the fact that this integrated system was able to retain old carbon, and not that much of that carbon was really new or entered the system since we started this experiment. So in fact, the new carbon appears to enter this label pool with a transit time of about 5 to 12 years, depending on which of the systems we were looking at. About 50% is lost during the first year, so it enters really very label pool. Very little of this label pool is going to be retained in mineral-associated organic matter. So the outputs are mostly explained by a high turnover of this label pool, which signal really very much resembles that of the atmosphere at the time, and the isotope signal of these outputs are very different from the mineral-associated organic matter, so that we can say that it's somehow not very much, this mineral-associated organic matter is not very much connected with the atmosphere, but the labile pool is the one that is fasting really cycling very fast. So the conservation of this oil organic carbon appears as the main mechanism to sustain this carbon in the soil in the integrated systems. However, to be able to retain this carbon in the soil, we need this fast pool that has a lot of inputs. So to conclude, physical fractions and isotope tracings are available tools that allow us to assess this carbon dynamics with a mechanistic approach. Unraveling these mechanisms of carbon cycling contributes to better understandings of carbon dynamics at the scientific level. We need this understanding to create mechanistic models that we can then trust, because we cannot measure everything and in every system. We need to model, but we need to develop these models under these type of frameworks where we can really have the scientific basis for this. And with that, I would like to thank you for your attention, and I'm open to questions. I'm sorry I went fast through some parts. I'm sorry about this cough that I have. And thank you very much.
Thank you very much, Virginia, and good recovery from the little episode. Well done. There is one question in the chat. Dear Virginia, could you find out if the old stable fraction of soil organic carbon has fire or biochar fractions in it?
No, but there's not a whole lot of biochar here in the soils that we have. We are starting in, well, our team at INEA is starting to test the role of biochar to be added as amendments, but we don't find a lot of biochar in our soils naturally. So.
Okay, thank you. No, I thought it was fascinating, and especially shows the value of long-term trials. You know, people 50, 60, 70 years ago had foresight to set these up. In New Zealand, we've got a couple of long-term trials. Pierre, do you know if similar type of work is being done? I'm thinking of Winchmore, which has been going for what, 70 years or so.
Yeah, so in New Zealand AgResearch, I think has been curating the Winchmore trials. I don't have in my top of my head the date at which it began. I think it's definitely more than 50 years old, but I can't exactly remember the year. I've never had a chance to work with the data myself, but that's by far our sort of oldest similar experiments here in New Zealand.
Yeah, no, it's about 70 years old, so 1955, I think they started. Yeah, just it might be interesting to do similar stuff as Virginia showed.
Yeah, it would, yeah.
Yeah. So we're sort of running out of time a little bit. I thought it was really interesting to have a sort of a global perspective on these technologies and ways of mapping. Does anyone else from the panel have anything they'd like to add? No? Okay.
One thing I can add to try and sort of synthesize across the different things we've seen today is that all these things can be linked. So we all touch on different things, but I guess in the future there's a lot of theoretical stuff to do in terms of, you know, trying to get spectroscopy to initiate some of these process-based model, for example, or use these approaches in mapping, you know, especially if you want to look at the future. This is where a lot of these process models shine. So, you know, I just encourage, I guess, people to not see that as different siloed themes, but more like little things that we are slowly trying to patch together.
Yes, Virginia.
Yes, I completely agree. I think it's from what I showed, the data that I showed always relied on dry combustion for soil organic carbon determinations ultimately as an ultimate step. But it's great that these tools are being developed more and more that we can rely on spectroscopy on after it's been well calibrated and all. To have a cheaper way of assessing soil carbon, so we can have more samples analyzed and in a faster way. It's a complementary approach, I think, of what we all mentioned today.
Yeah, the other thing I see the other value of spectroscopy is to go beyond carbon. So I guess we have this sort of flow to look at carbon in isolation of the rest of the soil system. There's a lot of interesting things to do if we could leverage spectroscopy to look at mineralogy, for example. I mean, here in New Zealand, we got, you know, a lot of allophanic soils. They have a very unique soil mineralogy that definitely plays a role in the nature of the organic matter in soils. So opportunity to link I guess the soil carbon stuff we've seen today with the pedology, for example, with soil formation.
So there's a question in the chat, sort of following on from my long-term trial. Pierre, how important is it to maintain trial sites like Ballantrae?
Yeah, so Ballantrae is another one. It's got a fresh new highway through it now.
It does.
It's
I I think it went to Parliament even to discuss putting that highway through this long-term site.
Yeah, no, it was, yeah. It's one of these things, Ballantrae, Winchmore, they're really critical, and I think they're getting more and more critical as the data comes in, right? So, and it's a tough one because they're always at risk, and we often hear of, how are we going to sustain Ballantrae or Winchmore? The value of long-term experiments only add up with time; they never go down. So, but you're right, Ben. It's something that's regularly at risk of losing funding, and because they're such long-term experiments, it's not always easy to fund these things. But what we see more and more, I think, throughout the literature, I mean, Virginia provides another example from Uruguay, is that as time goes by, the value of those experiments becomes really important.
Thanks for that. Felix, have you got anything to add before we close off? Because we're nearly on time here.
Yes, just a little advertisement. Next week, Wednesday at 19 p.m. is the next seminar, and this will focus on agroforestry. And that's it from me.
Okay. Yeah, well, thanks everyone for attending, and thanks especially to the panelists and presenters. Yeah, if you're interested in agroforestry, next week, Wednesday. So, okay, bye-bye from here.
Thanks, everyone.
Bye. Thank you.
Thank you. Bye-bye.